DNA, directly our next blockbuster. In the digital age of the Anthropocene, the bit has become the standard encoder of information, stored as binary code on computer hard drives. Yet the blind hand of evolution over billions of years has perfected its own hard drive. Robust, faithful in its copying mechanism, and with potentially petabytes of storage capacity per gram, DNA is the ideal repository of information. Yet it offers more than a preserved storage space, but offers the enchanting potential of recording and archiving its own major cellular life events. After all, the 4 base nucleotides are much more intrinsically linked with biology than computer hard drives. So the idea of DNA encoding has been around for almost 2 decades. Biblical verses, E equals MC squared, and even Watson and Crick's paper on the structure of DNA have all been encoded. Yet these involve manually transcribing into DNA and introducing this directly into bacterial genomes. But what if we could automate this process and exploit the full recording potential of DNA? Previous attempts at creating permanent genetic memory have yet to encode more than 1 byte of information. Yet coupled with the evolutionary optimized machinery of Crispr-Cas, we can manipulate cells to stamp the marks of their identity on their own genome. So this reality is still a fair way off, but the theory is there. As a proof of principle for DNA's recording capabilities, a team of researchers aimed to encode a short film into E. coli. Appropriately, the movie chosen was one of the first ever recorded motion pictures, 5 frames of a galloping horse from 1887. So all pixels were taken, split up, encoded into DNA across around 100 variant sequence blocks known as protospaces. These were electroporated into the E. coli with upregulated Cas1 and 2 proteins. Cas1 cleaves and Cas2 transcribes and inserts this into an array in order of appearance. So after cells grow, the metagenome of a population of cells is extracted, PCR'd at the CRISPR locus, and the original sequence is re-identified and translated back into the moving picture. First, they tried their techniques on a simple hand image. So the protospaces used have several key features to enhance the integration, efficiency of integration. So each base was exactly 33 base pairs long, which is the optimal length for Cas2 to integrate. And they used 2 strategies to test the effectiveness of encoding. The first, which is the rigid strategy, is where each base encodes for 1 of 4 shades of grey, while the second, the flexible strategy, is where a triplet codon encodes for 1 of 21 shades of grey, with some redundancy in the codon similar to amino acids. So the methods that follow apply to both of these. So for maximal biochemical stability, each protospace had around 50% GC content and no mononuclear repeats. This is where their redundancy plays in to make this more achievable. Secondly, at one end of the protospacer is 4 to 5 bases, making up a pixet. Now this is a coordinate separate to the pixels, which encode the colours. So here, each letter translates to a binary code which represents a number. For example, CGCG translates to 00110011. If you're unfamiliar with binary code, essentially each digit position doubles its value from the one before, and you add them all together. So this reads as 1 plus 2, skip 4 and 8, plus 16 plus 32, skip the last 2, which gives us 51, making up the ID coordinate. The first number 5 is how many pixels from the top, 1, 2, 3, 4, 5, and the second one is how many pixels from the left, so 1. So this encodes the position of pixel 1, and from this, a preset pattern determines the location of all the remaining pixels which were encoded by the triplet codes. Lastly, at either end of the protospacer is an AAG and a GA motif, respectively, which operate as the recognition sequence for Cas1 complete and to maintain the correct orientation of the insert. So thousands of these protospaces were electroporated into those upregulated E. coli. So the cells were allowed 7 days to grow, and after which they were PCR'd with primer specific for an invariant CRISPR leader junction. So after around 650,000 reads of the sequence, they were bioinformatically compared with the original genome to find those newly acquired spaces. And then these genomes were then translated back to the moving picture. So the rigid strategy gave an accuracy recall of about 88%, whereas flexible strategy yielded 96%, and so was chosen to use for the movie. So this clearly works for a single image, but there's a problem in how to scale this up to a moving picture. As there are 5 frames, there would be 5 variant protospaces with the same pixet code, which remember encodes the positional coordinates. So how can we tell from which frame each protospace is from? Crucially, they fed the bacteria 1 frame per day over 5 days. Now, as I said before, Cas2 integrates in order of appearance, so each newly acquired spaces inserts directly adjacent to the leader sequence, displacing the sequence applied beforehand. So the proximity of the spacer to the leader sequence denotes its pixel value, with frame 5 being electroporated most recently, being closest to the leader sequence. So after about 1 million reads, movie was recreated to 90% accuracy, and this here is the result.
In terms of the advantages of the study, the distribution of the data across a population prevented each bacterium from becoming overloaded with superfluous DNA. Also, it was extremely rare for a previously acquired spacer to be deleted, only 0.096% at the time. Additionally, no code for the frame order of the movie was required to be inserted into the protospacers. This had two key advantages, in that not only was the 33 base pair limit imposed on the size of the spacer not wasted, but it more crucially proved the robustness of the technique, since timing information was not needed to be encoded. However, there are some disadvantages. The technique demanded meticulous design and planning, and an optimal optimal protospacer needed to be troubleshooted. Despite this, they still used too many variable recognition motifs, meaning the technique was not as efficient as it could have been. Regardless, the potential storage capacity of the system is what is so enticing. DNA is so small that the world's data would weigh about 1 kilogram. An earlier study by the same team determined 7 times 10 to the power of 243 possible states could be encoded by the size of the spacer that was equal to that used in the movie. This is more than the number of atoms in the universe, so a more cautious estimation falls between 29 and 101 bytes. Now we've learned that it's essentially taken 1 day to photocopy an image, and it's in black and white. And it's taken about a week to watch a movie reel that's less than 1 second. So that's worse than trying to buffer an episode of Game of Thrones. So putting time and effort into this area seems ludicrous for the technology we have, so you may be thinking, what's the point? Well, it's not to store movies or put Wikipedia into bacteria. Rather, the Crispr adaptation system lays the foundation for an in vivo, multimodal, intracellular recording device, like a sort of dynamic living hard drive. Memories could be acquired and deposited into the genome over time, as information provided by researchers externally in the form of synthesized DNA. Or in the future, the hope is to connect the system to any cell type and then record information from the cell's own experiences and then play back its memory. Current methods involving Zyme recorder genes fail to show the dynamics of lineage between cells. Whole genome sequencing has the potential to trace lineage, but it is highly impractical across an entire multicellular organism. Conversely, the Crispr adaptation system makes this a possibility. This cell can do your dirty work for you, recording information into its genome rather than scientists having to painstakingly watch them. This could prove potentially vital in analysing the progression of Alzheimer's. As a non-destructive method, there is no requirement for the cells to be killed while the experiment is ongoing, and only a subset of the population needs to be sampled to retrieve the sequence information. For example, if a future study aimed at tracing all the decisions a differentiating neuron makes, from an early stem cell all the way to a highly specialised type in the brain, then neurological development would not need to be disturbed in the retrieval process. This would explain how you could generate cells for regenerative therapy, disease modelling, and drug testing that would all be better informed. So experiments last year introduced CRISPR-Cas9 into dividing zebrafish cells and tracked the mutations that occurred in daughter generations. The starter embryonic cells were marked with a barcode 300 base pairs long. Then 10 lots of the 20 base pair sequence within the barcode were targeted by CRISPR-Cas9. Once the experiment was over, the researchers then sequenced the genomes, and you were able to reconstruct lineage trees based on the unique pattern of edits each descendant cell accumulated. So these are all the potential uses that you could generate for it. Some wacky ideas could even include conducting cellular espionage by encoding secret messages into microbes. Or you could encode your own name into something else. It's like a form of genetic graffiti. Whilst all these possibilities are a long way off, the study took the first necessary step in demonstrating that cells can indeed store meaningful amounts of information and offer the potential as an in vivo recording device. Humans are developing the capacity to write their genome in the future, but I'll leave you to ponder that this forms the basis of a philosophical paradox. Can an intelligent being comprehend the instructions to make itself?